Hi, this is podcast number 10. And in this podcast, we're going to start talking about the rules of genetics. I can tell you that in 24 years of teaching principles of biology, this is typically the part of the class that students like the most because these are, unlike other parts of the class, these are pretty hard and fast rules and pretty interesting and hopefully easy to understand. And then also look at some data from pea plants to try to get an idea of how traits are inherited from one generation to the next. And in our map, we are at the organismal level here. In chapter 3, we'll run into organisms again in 6, 9, 12, and 15. And then, of course, you'll see more organisms in Principles 2. Here is Gregor Mundell. He is a monk, lived in Austria. He was interested in science. He was interested in patterns of inheritance, and he had some time on his hands. And so he used the pea plant as his so-called model organism. So a model organism is something that a scientist uses to study a general phenomenon, and the general phenomenon here was inheritance, how traits get passed on from one generation to the next. And even though we will talk about genes and genotypes and phenotypes and that sort of thing when we talk about Mendel's experiments, Mendel didn't have any idea about genes. He knew that there were factors, which is a fudge word that scientists use when they don't know what is involved, but didn't have any idea about genes. And but even so, he took really good notes. He had a really good system. The pea plant was not a great in terms of how long it took to generate, but he had some time on his hands, and he could perform these experiments over the course of years, and he could follow characteristics that he didn't need any fancy equipment, didn't need microscopes or anything like that. And one advantage of his organism is it had both male and female parts, and it didn't move. So he could easily say the gametes, the sexual cells from this male individual and that female individual, and you take pollen from the male parts and put it on the female parts of any two plants he wanted, and then he could control the breeding and then see what happens. So on a pea plant that might be in your garden now, there are both male and female parts on that flower, and the flower can do something called self. They can pollinate themselves, take sperm from their own pollen from their own self and use it to fertilize themselves on their stigma. Or you can do some pea surgery and remove either the male or the female parts and control who mates with who. And that's a big advantage in trying to figure out some of the things that he was trying to figure out. So in a pea plant, like you might have in your garden, they have certain characteristics. The stalk can be tall or short. Flowers can be different colors. Their seeds can be different colors. They can be round or wrinkly. And these are traits that Mendel followed over generations of pea plants. On the pea flower, there's both male and female parts. The male parts supply pollen to the female parts, and then the pollen lands on the sticky part of the female part of the flower, and then it grows a pollen tube that fertilizes the egg inside this part of the female part of the plant, and that's self-pollination. But you could also provide pollen from another individual plant, and so on. Here's a picture of those pollen tubes growing down the style of the plant from the stigma. Okay. And once the plant is fertilized, it'll make several peas, which we eat, but those are the seeds of the pea plant. And they have characteristics that you can follow, like different colors, yellow or green, and different surface characteristics, smooth or wrinkled. In his first experiments, he looked for what he called true breeding traits. That no matter what he did, this sort of pea would always make this kind of plant, and each pea would make one plant. So he found several of these true breeding traits, and he took 2 years to actually figure out this part of the experiment, and that he would not get variability if he knew what he was starting with in terms of these traits. So he took parents of these plants, or he took these plants with true breeding traits. In other words, a trait, a plant that always gave green seeds, a plant that always gave yellow seeds, and he did crosses between them, where he had the pollen from a green seeded plant landing on the female part, the stigma of a female plant that had yellow seeds, and then the reverse sort of cross also. And then saw what happened. And he called these plants the parental generation, or the P generation. No, not PEA, or the letter P. And then these had offspring in the form of peas. And if the male supplied green or the female supplied green, it didn't matter. All the offspring were yellow. And these were called the first filial generation. That's where the F comes from filial. And this is the first generation of offspring. Then he did a second set of experiments where he took those offspring of the parental generation. So he took the F1 peas, grew those up into adults, and then took their pollen and crossed it with each other. So these are so-called the brothers and sisters from this generation, crossed with each other, grown up and crossed with each other. So these are different individuals in the parental generation, not related to each other, but in the first filial generation, that cross, everybody is related to each other, brothers and sisters. Okay? So this is not how we would do a genetic experiment, definitely in humans, for sure. But in plants you can do this, and it reveals some of the rules by which genes are inherited. Okay, we took those F1 peas, crossed them with each other, okay, and he got peas produced called F2 peas. So this is after the parental generation, and then after the first generation, this is the F2 generation, second filial generation. And if you remember, in the F1 generation, everybody's yellow. All the peas are yellow. But in the F2 generation, some of the peas are green. So some of the characteristics of the pea generation, the parental generation, where one of the parents had green seeds, shows up again in the F2 generation. So how did that happen? Okay. If you look at some of the data, so 5 true breeding green plants. So these are plants that if you bred them with themselves, they always gave green seeds. And that's what true breeding means. And again, Mendel took 2 years to find that sort of thing out. And these pea plants, if you bred them with themselves, they had yellow peas and they always produced yellow peas. Now, if you crossed the ones that always produce green seeds with the ones that always produce yellow seeds, you get the F1 generation, and they're always yellow. And it doesn't matter if the mom has green seeds or if the dad has green seeds, always, 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 all the peas are yellow. And he called that trait dominant because if there's 2 traits here, a yellow trait and a green trait, and when those 2 traits are mixed in a breeding and all the peas come out yellow, yellow is considered dominant over green, and green is considered recessive. So here are some data that said that when he did this, he got 273 green or yellow peas and no green peas in the F1 generation. Okay. Now takes those some of those 273 yellow peas, and he self-crosses 258 of them. So the peas grown up to plants, and then they make pollen, and they have female parts, the stigma. They put the pollen from themselves on the stigma of themselves, and they cross with themselves. So these are brothers and sisters crossing with each other. Okay? And if you look at the ratio of the peas he got from that cross, he got about 2,000 green peas and about 6,000 yellow peas. And that's a 1-to-3 ratio of green to yellow. So again, yellow is dominant, green is recessive, but green shows up in the F2 generation and was missing in the F1 generation. So how did that happen? How can we explain that? It wasn't just in pea color. He also looked at other characteristics, true breeding characteristics of these pea plants. He actually had 7 different traits, looking at flower color and the length of the stem. But just looking at one other one to show you that it works the same way, he took a true breeding pea plant that had wrinkled seeds, that this plant, when it crossed with itself, always produced wrinkled seeds, and this plant, when it produced with itself, always had smooth seeds. And the F1 generation, similar sort of thing in that everybody was smooth, nobody was wrinkled, so smooth would be dominant over wrinkled. Wrinkled is recessive. Then cross these among themselves, self-pollinated these plants. Then he got, again, a 3-to-1 ratio, 3 times more dominant than recessive. So it works regardless of the true breeding trait, that you get this 3-to-1 ratio. However, if you notice on these tables, that's a lot of peas. 5,000, 1,850. If we go back here, 6,000, 2,000. Those are pretty big numbers. What if your numbers are not so big? So in 10 different matings, if we look at some of Mendel's data, that the individual ratios in a plant number 1 cross with itself and plant number 2 cross with itself, and you count up the peas, sometimes it's not 3-to-1. Sometimes it's less than 2-to-1. Sometimes it's greater than 4-to-1. So there's variability in what the number you get. And it's not until you get a really big number where you get 3-to-1 ratio. Okay? So 3-to-1 is the average probability of dominant over recessive, but in any individual mating, it might not be that. Just like the chance of any couple, human couple, having a girl is theoretically 50-50. But you probably know families where they're all girls and there's three girls in a row. So you need to have 1,000 girls or 1,000 kids to approach the 50-50 ratio that is theoretically what you would predict. And I'm not recommending that. Okay. And here's another situation. If we look at different traits, again, this is yellow instead of wrinkled, P color instead of P shape, and same sort of thing in that overall you get the 3-to-1 ratio, but any individual mating you might not, and it could be pretty different from 3-to-1. Okay. So now what happens in the F3 generation? Okay. So the F2 pays, 1/4 of them were green, 3/4 of them were yellow. That's their phenotype. That's what they look like. But what else can we learn about these P's? So if we cross these P's with themselves, they are true breeding. You always get green P's. Okay, so all the green P's are true breeding. But if you cross the yellow P's from F2 P's with themselves, some of those will always give yellow P's, but some won't. Okay? So about 1/3 of the yellow P's will give you always yellow P's, but these guys here, 3/4 of them won't. Okay? Why is that? All right. A new term. For any trait in a eukaryote that has two parents, okay, one male and one female. Two parents could be themselves, okay, but the idea is that there's a male part of the flower that supplies a part of the characteristic, and there's a female flower that supplies part of the characteristic. The male flower, part of the flower supplies pollen, and the female part of the flower supplies the egg. Each one of those contributes like 1 vote toward what the pea is going to look like. And if both of those traits are the same, okay, both of those genes are the same, both of those votes are the same, they're called homozygous. In the case of Mendel and his peas, the typical way to draw these, if it's a recessive trait, use a lowercase letter, like this is a lowercase y, and if it's a dominant trait, use a capital letter, like a capital Y. So in this case, where it's both genes are the same on the 2 chromosomes that code for that have a gene that code for seed color. One came from the male part of the flower, one came from the female part of the flower. They both are the same version, and if they are both the same version, they're called homozygous. And because it's a lowercase letter, it's called recessive trait. And when a homozygous recessive is supplying the gametes and it's crossing with another homozygous recessive, the only thing you can get out of that is homozygous recessive. So even if this was crossed with another green pea, that other green pea would have 2 little y's also. So if each of the 2 green pea plants supplies half of what's going to provide the next generation in F3, they're both supplying a little y, and so all their kids are going to be little y, little y. So each kid has to have 2 versions of each gene. Each of the parents had the same version of the gene, and so all the kids are going to be little y, little y. Okay, and they will all be green. All the homozygous recessive trait. If you have 2 versions that are both the same and they're both dominant, it'll be a different color. It's also called homozygous. And same sort of situation in that if you cross this with 2 parents that have this situation, each parent can only contribute a big Y because that's all they've got, and all the kids will be big Y, big Y. One came from one parent, one came from the other parent. They're both the same thing. They're going to be yellow no matter what. Okay? And those are true breeding. But what happens in a situation where you have a heterozygous plant? Okay? In a heterozygous plant, on one chromosome you have a little y, and on one chromosome you have a big Y. And when they are crossed, some of those will end up with little y, and some of those will end up with big Y, little y. And you can use something called a Punnett square to figure that out. Okay, so how do you do a Punnett square? Okay, let's say these are the 2 parents, and instead of phenotype, what they look like, this is a genotype. What genes do they have? So for this one trait, both parents have one homozygous dominant, yellow, it's called a version of a gene or allele, okay? And one has, and the other version of the allele that they have is a is a recessive allele, the green allele. So they have one yellow allele and one green allele, and both parents have that, okay? So if you figure out all the possible kids that these 2 parents could have, well, on the mother side, she could supply either the big Y or the little y to her egg. And on the father's side, he could supply either a big Y or a little y in the pollen. Okay? So the individual that results of that has to have 2 of each, but the gametes, the eggs and sperm that they make, will only have 1 of these. Okay? It'll either have a big Y or a little y, and this will either have a big Y or a little y. Then if you say, what happens if this egg is crossed with that pollen grain, then it would result in 2 big Ys. Big Y here and a big Y there. What about this egg and that pollen grain? That would have a big Y and a little y. The convention is always to put the capital letter first. Okay? And then this egg and that pollen grain, big Y, little y. And then this egg and that pollen grain, little y, little y. Okay? These are the 3 different kinds of genotypes. You can either have homozygous dominant big Y, homozygous recessive little y, or if you have 1 of each, that's called heterozygous. Okay? And these guys would be yellow because yellow is dominant over green. These guys would be yellow because you have 2 versions of yellow. And these guys here would be green because you have 2 versions of green and no yellow. So here's where you get that 3-to-1 ratio. You have 3 yellows, okay, yellow, 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 and only 1 green. So the 3-to-1 ratio that Mendel saw when he was counting out 6,000 peas, that came from this idea, and that there are 3 ways you can produce an individual that will be yellow, and only 1 way you can produce an individual that is green. Like that.